Hello everyone, hope you're all doing good. Myself Sanuja and welcome to a very new session of biology. So we were discussing the first chapter of plus 2 biology, sexual reproduction in flowering plants, right? We've already discussed a lot of topics in sexual reproduction and we've reached pollination, if you recall, right? We have discussed about wind pollination, water pollination, right? There were two abiotic factors of pollination or abiotic agents of pollination. And now the topic that we'll be discussing is pollination by biotic factors. So what do you mean by biotic factors? Mainly when we talk about biotic factors, we'll have to talk about animals, we'll have to talk about insects, right? So that's what we'll be looking today, right? So here, this is where we stopped last day, right? This is the slide that we were watching in the last class that we ended the class with. Both wind and water pollinated flowers are not very colorful and do not produce nectar, right? So this was a statement that we discussed in the last class and that's where we ended the class, right? So now moving on, we will have to learn about pollination by animals, pollination of flowers or pollination of plants by animals or by biotic factors, by living factors, right? So when it comes to animals, a lot of animals, okay, a number of animals pollinate flowers of different species. So which animal pollinates a particular flower totally depends upon which species it is, okay. So the first one and the most important one obviously is bees. Right, bees, we all know, honeybees, right, we've all heard about bees, we have all seen honeybees, like some may have like even got a bite, right, from honeybee, not like a sting. And then you have butterflies, obviously, we have seen butterflies, right, we have seen butterflies roaming around flowers, so butterflies obviously. And then you have flies, beetles, wasps, ants, moths, and obviously birds, okay. You have a lot of animals to be exact, you even have some mammals and stuff, but anyways these are the most important. And you also have like bats, okay, bat is a mammal but still it pollinates plants, okay, it is an agent of pollinator, it's a biotic agent. And then you have some primates like lemurs, okay, you have lemurs, some primates, they also pollinate flowers. And you even have reptiles, okay, even the lizard, the gecko lizard, you're not seen, maybe you're not seeing a gecko lizard, it's not like the normal lizard that we see, it's kind of a different lizard, but anyways gecko lizard and garden lizard and all this pollinates flowers, okay. But the most common out of all this is bees, right, that's quite obvious, okay. Now moving on, here you can see a picture of a bee pollinating a flower. Now, we've already learned the features of a flower that is pollinated by wind and the features of a flower that is pollinated by water as well. Now let's look at the features of a flower which is pollinated by an insect, okay, like bees and like in animals in general, okay. Let's look at the features of a plant that has to be pollinated by insects. So before looking at the slide, just think, okay, just use your brain and think, what would be the features of a flower? I mean, an animal or an insect have to get attracted to the flower, right? At the beginning, the insect has to approach the flower, and in order to approach the flower, it should be attracted to the flower. So obviously the flower should be, like the insect should be able to see the flower, right? So of course it should be large, right, it should be large because the insect that's coming should be able to see the flower, right? So it should be large, it should be colorful, like it should attract the insect towards it. So it should be colorful, like red flowers, you have blue flowers, you have pink flowers, you have a lot of variety of flowers, right, variety of colors. So it should be colorful obviously, and then it should have some kind of fragrance, okay. This fragrance can be good or bad, depends upon the species, but anyways it should have some kind of fragrance. Of course we've all picked a flower and we have all smelled it, right, and of course it smells so good. Some flowers smells so good and some flowers doesn't. Anyways, fragrance is an important part or an important feature of a flower or of a plant which is pollinated by an insect, okay. Now it should be rich in nectar. What do you mean by nectar? You know what nectar is, right? It's like honey or nectar, we call it in different names. It's basically the reward that you give the insect, right? So the insect doesn't know that it's gonna pollinate or anything. Insect actually is not even conscious whether it's pollinating or not. Insect basically comes to collect nectar. That's the basic role or basic agenda of an insect, right? And meanwhile that happens, insect automatically, unknowingly or knowingly pollinates the flower. That's what usually happens. I've already told you about insect pollination before. So that's what happens, right? So obviously there should be nectar, right? The flower should be rich in nectar. It's only then the insect will come to the flower, right? So it should be rich in nectar. Also, if the flowers are small, just imagine if the flowers are small, it will not be easy for the insect to see it, right? Insect will be flying, and while the insect is flying, it should be able to see the flower, right? As I already told you. So either the flower should be large, or if the flower is small, then it should be in the form of some groups, right? See, imagine I'm drawing a flower here, okay? So this is kind of a big flower I'm drawing, right? I'm not good at drawing, but still you can see a big flower here, right? And if this is colorful and if this is beautiful and all that, an insect can easily see this flower, right? Insect can easily see this flower. But if you have a flower like this, okay, if you have a flower like this is a small flower, and it will not be easy for the insect to find this flower as easy as it was, like to find this one, right? But if these small flowers are found in groups like this, if they are found in groups like this, and if they are colorful and beautiful and all that, the insect can easily find. So you'll have multiple flowers in the form of a group, and then it will be easy for the insect to find it, right? So either it should be large, or if the flower is small, it should be in the form of groups. So what do you call a group of flowers? A group of flowers is usually called an inflorescence, right? It's called an inflorescence. So either the flower should be large, or the flower should be in the form of an inflorescence. Okay, the next point that I have to convey is that I've already told you that fragrance is an important part, right? But the fragrance can be good or bad, right? In the case of flies and beetles, they're actually attracted to foul odors. Okay, they're not attracted to good smell or fragrance, but instead they are attracted to foul odors. Okay, something that's not that pleasant for us to smell, something that's foul, something that we don't like. Okay, it's easy to remember, because where do you see flies? You see flies in places where you have waste and stuff like that, right? So obviously flies and beetles get attracted to foul odors. Okay, it's not getting attracted to good odors or a good fragrance, but instead it is attracted to a foul odor. Is that clear? So that's what you see here. Then as I already told you, nectar is a reward that we give the insect, right? So the insect is pollinating the flower for us. Imagine I am a flower, so insect is pollinating me, right? Or if I am a plant, insect is pollinating my flower. So insect is doing some help to me, right? So I should also do some help, or I should also give the insect some reward, right? So what do we give as a reward to the insects usually? The one thing that we give is, of course, the nectar, right? As I already told you. In other cases, you also give the pollen grain, which is actually not necessary for the insect. It's not necessarily a reward. But if you are asked what floral rewards are, if you are asked what floral rewards are, you should obviously write nectar and pollen grain, because nectar is, of course, a pollen reward, or if it's a reward for the insect. That's obvious. But also write pollen grain. Okay, even if it is not useful for insects, still go and write that. Okay, and of course the process of insect pollination. Haven't I told you the process already? The insect gets attracted to the flower because the flower is, of course, large, colorful, or maybe in some cases in the form of a group, right? So the insect will obviously be attracted to what? The flower, right? So insect gets attracted to the flower and moves to the flower, and of course there will be nectar, right? So where do you actually find this nectar? Where is nectar is actually present? That's the question, right? Okay, just look over here. If this is a flower, okay, I'm drawing a flower right now. Imagine these are the petals or something, and then you have the ovary. I hope you remember how an ovary looks, how a female reproductive system looks, and how a male reproductive system looks. I've already discussed all that in detail, guys, so I hope you remember all that. Anyways, we have the ovary, and then we have the style, the stigma, right? So there goes the female reproductive system, and then you have the male reproductive system, which includes stamens, right? So imagine you have multiple stamens over here. You have multiple stamens over here. Is that clear? Okay, you have multiple stamens and anthers and all that kind of stuff. So there goes the male reproductive organ. So this is a flower, and it's so colorful, it's so large. Now what happens? An insect gets attracted to it. So the nectaries, or the nectar, is actually in most cases hidden below the ovary, somewhere over here. Okay, so the nectar is usually stored in some kind of nectaries or something. So the place where nectar is stored is called as nectaries. Anyways, it's not important. Just know that the nectar will be placed down here, below the ovary. So what will the insect have to do? What will the insect have to do? Okay, I'll change the color of my pen for representing an insect, So this, imagine, is a butterfly or some kind of insect or something, okay. So the insect gets in. to the flower and it has to find the nectary. So the nectar is obviously at the bottom, right? So the insect moves towards the bottom, and while it moves, okay, while it moves towards the bottom, obviously there will be pollen grains released over here, right? From the anther there will be pollen grains released. So unknowingly or knowingly, the pollen grain accidentally obviously gets to the body or gets onto the body of the insect, because the insect obviously touches the anther and that's how it moves down, right? So the pollen grain, where will the pollen grain fall? The pollen grain will fall on the body of the insect, and the insect may or may not know this. It's not important. Anyways, the pollen grain falls on the insect, and the insect goes down. It collects its nectar. Okay, it does its duty, and it goes back. Okay, it goes out. It has to go out, right? So it goes out. Okay, so now the insect is going out. So this insect that goes out of the flower has pollen grains on it, right, on its body, and when it enters another flower, so a different flower will also have almost the same kind of representation, right? So if it enters another flower, that pollen grain, this pollen grain that the insect has on its body will be developed on the stigma of the other flower, right? So that's how the process of insect pollination goes. It's basically common sense. It's basically something that we can imagine or something that we can kind of see, right? So see, like imagine in our mind. So it's not anything complicated. So these were the features of a flower pollinated by insects. Okay, I hope that's clear. Now we were talking about floral rewards, right? So the main two floral rewards were nectar, right? Nectar, and what was the second one? The pollen grain, right? So the nectar and the pollen grain. These were the two rewards that we have already talked about. But there are other floral rewards as well. What are the other floral rewards? The first one is a safe place to lay egg. Okay, in some cases the insects need a safe place to lay its eggs, because the insects also need to reproduce, right? So many insects reproduce by laying eggs. So the eggs should be safe and protected, and if it's inside a flower, of course the egg is protected, right? It will not be affected by wind, water or anything. It will be completely protected if it's inside a flower. So usually in some cases insects put their eggs inside a flower. So what does the flower has to do? The flower does not take any extra efforts. The flower can just stand there and just protect the eggs that are inside the flower, right? That's what the flower has to do. So it's actually like a symbiosis or something. I mean, the flower is also getting benefited by the insect pollinating it, and the insect is also getting benefited by storing its eggs inside a flower. So the other floral rewards that we were talking about, the first one is a safe place to lay eggs. Okay, it's usually inside the flower. If I give you an example, you have a family Araceae, and in that you have a genus called Amorphophallus. Okay, it's a genus called Amorphophallus, and in some plants, in some species of Amorphophallus, the flower will be so large or so tall. Again, here you can see the word tall here. So if the flower is so tall and if the flower is large, then obviously the insect can lay eggs inside it, right? So in some cases of the genus Amorphophallus, you can see that insects actually lay eggs inside it. Okay, another example is Yucca. Okay, so Yucca is basically a genus of a plant and a species of moth. Okay, so you have Yucca, which is a plant species, and you have a moth. Okay, so this Yucca and moth is dependent to each other. Okay, they are dependent. So what happens is that the Yucca and moth, they cannot complete their life cycles without each other. So they are dependent. Okay, so the Yucca, which is a plant genus, and another moth, which you don't have to study the name. You have a moth, and this moth and this Yucca plant actually depends on each other. How do they depend on each other? Let's look at that. The moth actually deposits its eggs in the ovary of the Yucca plant. So you have the Yucca plant, and inside the Yucca plant you have an ovary, right? And inside the ovary is where this moth lays eggs. See, the moth deposits the eggs inside the ovary of the plant. And how is the flower benefited? Obviously the flower is getting pollinated by the insect, by the moth. Okay, so I hope it's clear. So that's an example of how plants and insects actually have kind of a symbiosis relationship, or how they help each other, right? And this comes under the heading floral rewards. I'll show you a picture even. See, so this is a picture of a moth pollinating the Yucca flower. Okay, this is actually the Yucca flower, and this over here, the white colored thing is the moth that you're seeing. Okay, so that's about insect pollination, right? Now the next topic, so the pollination is over right there because we've already discussed about abiotic factors or abiotic agents, and now we have discussed about biotic agents as well. You don't have to learn every other biotic agents in detail, okay? You have to only learn about general stuff like, you know, about insects in general, okay? You don't have to go like deep into birds and bees and flies and beetles. Just remember, flies and beetles get attracted to foul odors. That's the only point that you have to specially remember. All you know, the rest of it is kind of obvious, right? Okay, so the next topic that you have is outbreeding devices. What's the next topic? Outbreeding devices. What do you mean by outbreeding? You should know that first, right? So what do you mean by outbreeding? We've already discussed about different types of pollination, right? Self-pollination, cross-pollination and all that. We have discussed about all that. What will happen if you have repeated self-pollination? It's a question for you. Have you got the answer? If, you know, repeated pollination or repeated self-pollination take place, it may lead to something known as inbreeding depression. What is inbreeding depression? Imagine there's a plant A, okay? Just imagine there's a plant A and there's a plant B. Okay, there are two plants, but this B doesn't have a role here because we are talking about self-pollination. So just concentrate on A. So this A actually has many good characters, but it also has some bad characters. Okay, it also has some bad features. Maybe it may not, you know, be a plant giving good yield. Maybe it is giving bad yield. Okay, just let's take that example. So A is a crop plant, but it has bad yield. It's giving bad yield. So if we self-pollinate it, okay, so in self-pollination you don't have two parents, okay, that's why I cut the B. You don't have two parents, you only have one parent over here. So you have A, and from this single plant after self-pollination you get another plant known as C. Let's take it C. Okay, so C is a plant that is formed from A by self-pollination. So whatever bad characters are there in A will obviously move to B as well, right? Sorry, to C as well, right? Just imagine, you don't have any kind of genetic mixing going on. So simply saying whatever bad characters and whatever good characters were in A will obviously be automatically passed on to C as well, right? So if A is a plant that's not giving good yield, obviously C will also become a plant that's not giving good yield, right? Just use your common sense. That's what will happen. So after generations and generations, this bad character starts to get accumulate, or it accumulates. Okay, gradually it accumulates and the plant or the crop turns into a bad plant with a very bad yield, and which is not useful at all. So this is known as inbreeding depression. This is known as inbreeding depression. So what do you mean by inbreeding depression? If there is a bad character in a particular plant, and if we do repeated self-pollination on it, the generations that follows will have or will accumulate the bad characters of the parent, right? So that's what is known as inbreeding depression. So this is a very big problem, right? So when is inbreeding depression happening? When we do inbreeding, right? When we do inbreeding or when we self-pollinate it. So self-pollination should be prevented somehow. So that's our goal, right? Self-pollination should be somehow prevented. So how do we prevent self-pollination? Or in other words, cross-pollination should be promoted, right? Or in other words, outbreeding should be promoted. What do you mean by outbreeding? Simply cross-pollination, right? So cross-pollination should be promoted, or you need to have some devices, or there should be some methods to stop or prevent self-pollination and to promote, right, or support cross-pollination. So what are the ways? Or it's basically not any device as in they're not physical devices. They're just methods, some methods that a plant adopt to prevent self-pollination. So can we look at it? Can we look at all the 4 methods that are given here? Right, look at the first method. Pollen release and stigma receptivity is not synchronized. What do you mean by that? Pollen release. So where do pollen release from? The male reproductive organ. And what do you mean by stigma receptivity? I'll tell you. Usually a pollen should be mature enough to be released. And that's something we already know. A pollen should be completely developed, or should be in 2-cell stage or 3-cell stage in order to release. Cool. And the stigma should also be ready to receive the pollen. If the stigma is too young, it will not receive the pollen. Only if the stigma is ready to receive, or if it is receptive, will it accept the pollen grain. So it's not like the pollen grain can be released any time after its maturity and the stigma will accept it at any time. No, that's not what happens. What happens is the pollen release only happens when the pollen is mature, and the stigma only accepts the pollen if it is ready. Okay, the stigma has to be ready in order to accept the pollen. So these two processes, the pollen release and the stigma being ready, or the stigma receptivity, should be at the same time. Only if it's at the same time will pollination actually take place, or will fertilization take place in the later stages, right? What do I mean by that? Imagine you have a pollen. So imagine you have a stigma. So this is a stigma that I'm drawing, and you also have a pollen. You have a pollen coming, right? So this pollen is totally mature. You have the exine and the intine. I hope you remember all that, right? The walls of pollen grain. Okay, you have the exine, you have the intine. The pollen is completely developed. It's ready. Okay, it's totally ready. But the stigma over here is not ready. The stigma is not receptive. So even if the mature pollen grain comes over here, it won't be accepted by the stigma, right? So this basically prevents self-pollination, right? So if this is happening in the same plant, and the stigma is not ready to accept the pollen from the same plant, okay, it's not ready, then what will happen? Pollination will not happen, right? So this is one way of how we can prevent self-pollination. This is one way. So what is the one way that we were talking about? Pollen release and stigma receptivity should not be synchronized. It should not be synchronized. It means it should not happen at the same time. It should happen at different times, okay? The pollen release and the stigma receptivity should not be happening at the same time, or it should not be synchronized. I hope you understand that. The next point we have here, the second point, is that the anther and the stigma at different positions. What do you mean by that? I'll draw it for you. Okay, I'll draw the whole figure for you. The anther and the stigma should not be at the same position, or at complementing position. So the anther and the stigma should not be at the same position. What do you mean by that? Imagine a flower, okay, with a female reproductive organ, the ovary, the style, and the stigma over here. And I'm drawing the male reproductive organ, or the stamen, okay, just look at it. So this is the stamen. This is the stamen. It's a single plant, okay, it's basically a single plant, and I'm drawing the male reproductive organ, or the stamen here. So this is the anthers that I'm drawing right now. So you have the anthers as well. Cool. So what will happen after maturity? So here it's not the case of not synchronized maturity or anything. Here both are mature. Okay, both are ready to, stigma is ready to accept the pollen grain, and the pollen grain is also mature. So obviously the pollen grain will be released. Right, the pollen grain will be released. Cool. But here's a problem. What's the problem here? The stigma is too at a height, right? Or it's too far away from the pollen, or from the stamen. So what will happen? The pollen grain will not be able to go into the stigma. The pollen grain, of course, as we have gravity, the pollen grain will fall downwards, right? The pollen grain will fall downwards, or even if it flies, it will fly in different directions, right? If it will, with the help of wind or insects, it will fly in different directions. It is very hard for the pollen to climb up to the stigma. It's very hard. It won't happen. Okay, so if the stigma and the stamen, or simply saying the female reproductive organ, the pistil and the stamen, are located at positions such that it's very hard for the pollen grain to reach to the stigma, self-pollination will be prevented, right? So that's your second point here. So what is the second point? The anther, or simply saying the stamen and the stigma at different positions. So here the positions are totally different, and it's not easy for the pollen grain. It's not at all possible for the pollen grain to climb onto the stigma of the same plant. Okay, so in this way also self-pollination can be prevented. So this is another outbreeding device. Okay, now the third one. What do you mean by third one? What is the third point? Self-incompatibility. Right? What's the point? Self-incompatibility. What do you mean by self-incompatibility? We'll look that in detail. Okay, I'll explain it to you in the next slide. Okay, just keep it in mind that self-incompatibility is also an outbreeding device. Okay, and in fact, it's a genetic mechanism. It involves something with genes or something like that. Okay, so it's a genetic mechanism, and it is also a very important outbreeding device. I'll discuss it in detail in the next slide. Okay, now let's look at the fourth point. Production of unisexual flowers. What do you mean? What do I mean by that? It's common sense. I've already mentioned that self-pollination only takes place in bisexual plants, right? I mean, if a flower only has the male reproductive organ, how do you plan to conduct self-pollination? It's not possible. Self-pollination only happens when the pollen grain moves from the anther to the stigma of the same flower. But if a flower is unisexual, if a flower is unisexual, it will only have any one of the reproductive organ, either male or female, right? So it will only have either one of the reproductive organ, and hence self-pollination will be prevented, obviously, right? So that's the fourth method. So you have simple methods. You have 4 simple methods over here in which self-pollination or self-incompatibility we'll be discussing right now. Pollen-pistil interaction. This is basically what self-incompatibility is. It's almost a same thing, okay? So I'll be discussing pollen-pistil interaction and self-incompatibility together so that you will get an idea, you will get a crystal clear idea of what self-incompatibility is. From the word self-incompatibility, what do you get? What idea do you get? Self-compatibility. We've all heard about compatibility, right? When we look for life partners or even friends, compatibility is really important, right? If we're not compatible, we may have, usual fights, and, the relationship may break later on. So compatibility is something that we often find, right? Often try to find between partners or friends. So compatibility is very important, even for plants. Okay, even for plants, compatibility is important. But here we are talking about incompatibility, which means the plant is not compatible, right? Incompatibility means that it is not compatible. But what do you mean by self-incompatibility? Let's see. Let me begin with pollen-pistil interaction, and as I end this topic, you will automatically understand what self-incompatibility is. Okay, pollen-pistil interaction. You know what a pollen is, right? You know what a pistil is. They are basically interacting. So over here you have to imagine an interview. Okay, just imagine an interview. You're going for a job interview. Okay, so you will have the interviewer and you will be the interviewee, and you will go sit there, you will interact, right? How will you interact? You will speak, right? You will use your language, you will interact with each other. The interviewer will ask you some questions, and if you answer good, and if they think you are capable, or if they think that you are the right person, they'll select you, they'll choose you for the job. So that's what usually happens in an interview. Cool. In the same way, the pistil also interviews the pollen. It's a simple way to remember this thing. Okay, the pistil or the female reproductive organ interviews or interacts with the male pollen. Right. So if a pollen from a completely another species come, what will happen? The pistil will obviously interact with the pollen, and here interaction doesn't take place with the help of language, because plants do not have language, right? It happens with the help of chemicals. Okay, the pollen grain will secrete some chemicals, and by identifying that chemicals, the pistil can understand whether the chemical or whether the pollen is from the same species or not. If it's from the same species, it will accept the pollen. Okay, but if it's from a totally different species, then pollination should not happen, right? So what will happen? The pistil will reject the pollen. So that's what actually happens in the case of pollen-pistil interaction. So this was an outline. Okay, this is basically everything. Okay, it's basically the whole idea, and we'll be discussing terms, and I'll be telling you how to write it in your exam, because you can't obviously write it like how I said right now. Okay, so what's the first step over here in pollen-pistil interaction? Pistil recognizes the right pollen. So the pistil only accepts the right pollen, as I already said. So if it's the right pollen, it will accept it, and if it's the wrong pollen, it will obviously reject it. We've all faced multiple rejections in life, haven't we? So this rejection is not a new thing for us. So if we are right, it accepts it. If the pollen is the right person, the pistil obviously accepts it, but if it's the wrong person, it goes on rejecting. And then what happens is, how does this rejection and acceptance happen? It happens with continuous interaction with the help of chemical components, as I already told you, right? Plants does not have a language, so basically they use chemical components to communicate or interact. Cool. If it's a compatible pollen, the pollen germinates. Otherwise, the pollen don't germinate. So what do you mean by germination? I'll draw a figure over here. Imagine this is the stigma, okay? And a pollen has fallen. 3 pollens have fallen. For that matter, right? 3 pollens have fallen. These are the 3 pollens. Let us name them A, B, and C. These are the 3 pollens that have fallen on the stigma. Over here, B is the only compatible pollen. B is the only compatible one. A and C are incompatible. Cool, understood. So how will it work? The B, as it is compatible, will germinate and form a pollen tube. Alright, and form a pollen tube and gradually reach the ovary and the ovule, and the process goes on. okay, but in the case of A and C, the pollen will not germinate. Okay, see in the case of A, the pollen did not even germinate, the pollen tube did not even emerge, and in the case of C, yes, the pollen tube did emerge, but the growth stopped right here, the growth stopped right here, it didn't grow further. Okay, so A and C were incompatible, and hence it didn't germinate, the pollen didn't germinate, and even if it did germinate, the pollen tube didn't grow. Okay, but in the case of B, it was compatible, and hence the pollen tube has grown up to the bottom, up to the ovary, right? So this is another point, right? So here compatible pollen is the only one that germinates, keep that in mind. Okay, so as I already told you, the pollen tube grows if it is compatible, otherwise it won't. So if it's compatible, the pollen tube grows and it obviously reaches the ovary. It obviously reaches the ovary. Now before explaining the last 3 points, I'll connect this thing that I've already told you, the concept that I've already told you, with self-incompatibility. How will I connect it with self-incompatibility? Look over here, you have a flower. So this is a single flower, I'm talking about just one flower over here, okay. So this is a single flower which is bisexual, okay, it's bisexual. What do I mean by bisexual? It has an ovary, a style and a stigma, that is it has a pistil, and it has a stamen as well. Okay, it has the stamen as well over here. Let me increase the size of the stamen. It has the stamen as well. Cool. It has the stamen as well. I have been repeatedly drawing the same picture again and again, but yeah, I have to explain things, so I'll have to draw. Okay, so this is the pollen grain, this is the stamen and the pistil. Cool. Now the pollen grain is released. The pollen grain is released from the let me make it a bit smaller because it's only then you can, okay, I hope you can see right now. I'll make it a bit small, okay. I'll make it a bit smaller. I'll make the ovary a bit smaller. Cool, you have the ovary, so it's a bit small now. Okay, so the pollen grains will obviously release to the stigma because self-pollination is happening over here, it's a single flower, okay. So the pollen grain which is released over here will be released to the stigma, onto the stigma. So you have the stigma over here. In the case of self-incompatibility, what will happen is the pollen, okay, of the same plant will be unable to germinate on the female part or the pistil or the stigma. Okay, what did I just say? I'll repeat it for you. The pollen grain of the same flower, okay, the pollen grain of the same flower to be precise. I can also use same plant, but let me be precise and use same flower. The pollen grain from the same flower will not germinate on the stigma of that flower, or simply saying the pollen and the pistil does an interaction, it performs an interaction obviously, but as it's from the same flower, it's from the same flower, and as we have to block self-pollination, as we have to prevent self-pollination as an outbreeding mechanism, the female flower actually, or the female part, the female organ, okay, or the stigma actually says, okay, you can't germinate over here. So this is what self-incompatibility is. So it's basically what happens is a pollen-pistil interaction itself. The process is going on as pollen-pistil interaction, but the interaction happens between the pollen and the stigma of the same flower, and in that way pollination will not happen, which means self-pollination will be prevented. This is known as self-incompatibility. I hope it's clear right now. Okay, so this is why I told you that I'll explain it later on. Cool, so that's all about self-incompatibility and pollen-pistil interaction. Okay, so let's continue from here. The pollen tube has grown, right? The pollen tube of the compatible pollen has grown and it has reached the ovary. From the ovary, where does the pollen tube grow? Where does the pollen tube go? The pollen tube has to grow and go somewhere, right? It has a name. So where does the pollen grain actually go? Let's look at it. I'll draw it for you, okay? So here, I'll be drawing the ovary part or the basal part of the stigma. So this is the ovary, okay. Sorry, you guys. So this is the ovary over here, right? You don't have to concentrate about the style and stigma anymore because that part is already over. We've already discussed it, right? So here you have the ovary, and inside the ovary you obviously know what's inside the ovary. What's inside the ovary? The ovule, right? Obviously, we have already learned about the structure of ovule. We have already discussed in detail the structure of the female gametophyte or the ovule. So you have the ovule over here as well. Cool. And now the pollen grain, right? The pollen grain is growing right now. Okay, the pollen grain or the pollen tube actually. The pollen grain germinates and the pollen tube is growing. Okay, the pollen tube is growing. And what do you find inside the pollen tube? What's there inside the pollen tube? Inside the pollen tube, you can actually find two gametes, right? You can find two gametes, 1 and 2. The thing in yellow is the gametes, okay? So you have two male gametes, right? So inside a pollen grain, there will be two male gametes, right? We've already discussed all that in detail. So you'll have two male gametes over there. So the pollen tube continues to grow, right? The pollen tube continues to grow. And where does the pollen tube actually go? So the pollen tube actually has a destination. What is the destination? What are we actually doing? We're doing pollination here, right? We have completed pollination, and now the male gamete should actually reach the egg, right? The fertilization is the next step. So in order for fertilization to take place, you need the male gamete and the egg at proximity, right? The male gamete and the egg should be close to each other in order for fertilization to take place. Clear? So where are we traveling? Where are we going? We are going to find the egg. So where do you see the egg? The egg is over here, right? So this is the micropyle end. I hope you remember what a micropyle end is. And this is the egg over here. You'll also have two synergids. Right? And you have the egg as well. Clear? So the male gamete or the pollen tube is actually going to find the egg right now. It's in a hunt. Okay, it's going to find the egg. Cool. So the pollen tube grows this way. And enters the ovule through the micropyle region, because as you already know, at the micropyle region you don't have integuments, right? So it can easily enter. Okay, the pollen tube easily enters the micropyle end, but after entering into the micropyle end, it needs some kind of guidance, right? It needs some kind of guidance to move into the egg. So who gives that guidance? That guidance is given by filiform apparatus. Filiform apparatus. Where will you find filiform apparatus? I'll draw it for you. Just look over here. Oops. Did I mess up? Just look over here. Okay. I'm totally messing up. Don't mind. Okay. So this is the ovary. I'll redraw it. It's cool. Okay. So you have the synergids over here, and in front of the synergids you have the filiform apparatus, right? So you'll have the filiform apparatus over here. I repeat, I'm not good at drawing, but I'll show you a real figure, okay? I'll show you the NCERT figure, so don't worry. Just try to, take the concept in. So in front of the synergids, you have the filiform apparatus, and that filiform apparatus, they are basically finger-like projections. They look like something like this. Okay, they are finger-like projections, and it's known as filiform apparatus. And this filiform apparatus actually guides the pollen tube or guides the male gamete towards the egg. That's what actually happens. So look over here. It reaches the ovary, the pollen tube reaches the ovary, and it enters the ovule through the micropyle, as I've already told you. And then the pollen tube enters the synergids through the filiform apparatus, right? So the filiform apparatus actually guides the pollen tube to the egg. Okay, I hope it's clear. So this is what actually happens. So what is the next process that should happen? The next process is obviously fertilization, right? Of course, the male gamete has reached the egg, and the only process left to happen is fertilization. After fertilization, reproduction is kind of over, right? So that's the basis of reproduction. Fertilization, basic process of reproduction is fertilization. So we were discussing pre-fertilization events over here, and the next process is fertilization. So pre-fertilization events are actually over. We are ending the section here. Pre-fertilization events is over. I'll end it with a figure. Okay, I'll end this portion, this heading, pre-fertilization events, with a figure. Okay, so you have a figure over here. Just look at this figure, okay, figure D. In this figure, you can actually see all that I've drawn before. I've drawn a very beautiful figure before, have I? No, I haven't. I'm very bad at drawing, and I've drawn a very bad figure, in fact. But over here, you can see that I've drawn. You have a very good figure over here. Okay, you have a very good figure. It's a figure from NCERT, and in this figure, you can actually see the synergid. Right, it's a zoomed image, so the synergid is actually quite big, and you can also see the filiform apparatus. As I've already told you, they are finger-like projections. So you have the filiform apparatus, and the filiform apparatus is what guides the, pollen tube. So this is the pollen tube that you're seeing, the pollen tube to the egg. So you'll also have male gametes over here. Okay, so this is a picture of what I was trying to draw. Okay, in the previous slide. Now I guess it's clear. Okay, you have seen a beautiful figure, and I hope it's clear. Okay, now let's move to the next topic. What would be the next topic? As I already said, it would be fertilization. But Just stick with me. I also have to tell you a small topic before going into fertilization. The small heading, or it's a very simple concept, and that concept is artificial hybridization. What is the concept? Artificial hybridization. What do you mean by artificial hybridization? What do you mean by the word artificial? Artificial is something that we do, right? Something that's man-made or something that we do is known as artificial. Okay. What do you mean by hybridization? I'll tell you. Okay. Just forget the slides. I'll explain it to you. Imagine there's a plant A and there's a plant B. Two plants. There are two plants, A and B. So imagine they are mango trees. Okay, it's my favorite example to explain with. So you have mango trees. Okay, two mango trees, A and B. The specialty of A is that it has very good yield. Okay, it has really good yield. Okay, the mango is really sweet. Okay, and the size of the mango is huge. Right, and you have a lot of number of mangoes. You have a lot of number of mangoes at a single period. Okay, it also flowers every year. Okay, it also flowers every single year. So by all means, A is a very good plant, but there's one single issue. What is the single issue? A does not have disease resistance. A is not disease resistant. So A is really susceptible to diseases. Okay, so it's a very good plant. It's a plant that we look for, right? It's a plant that we want. But the problem is it's not having disease resistance, or it is susceptible to diseases. It can catch diseases so easily and the plant can die. So that's the problem that's the story of A here. And now let's look at the story of B. In the case of the B, it's another mango tree. In the case of the B plant, you don't have good yield. Nope, you don't have good yield. There's no good yield, there's no yield at all. Maybe you will find 2 or 3 mangoes, that's all. So the number of mangoes is also very less. The mango is not at all sweet, it's actually sour. The mango is really sour, and mango is not big. Okay, mango is not even big. You'll get small mangoes, and it will not flower yearly. It will not flower yearly. So by all means, B is a very bad plant. Okay, B is a very bad plant. We don't need that plant here. But there's one benefit. What's the benefit, or what's the good point here? B is disease resistant. B is disease resistant. It's not susceptible to diseases at all. Okay, B is actually disease resistant. So what would we ideally do? We would wish, what would we wish? We would wish if A had the quality of B. No, A had the single quality, the disease resistance, right? If A had that, A would have been a better plant. A would have been an awesome plant, right? So in order to bring this one quality from B to A, we do a process called as crossing. Okay, crossing. We cross these two plants. What do I mean by crossing? So it's nothing but we take the pollen grain from here and we put it to the stigma of this plant. Okay, we put it to the stigma of A. That's what you meant by crossing. We actually breed them. So this crossing, the whole process is known as hybridization. What is it known as? It's known as hybridization because we are making new plants, and these new plants are called hybrids. So this process is known as hybridization. Okay, and we are performing it, right? Nature is not doing it on its own. We are performing it, and hence it's known as artificial hybridization. So now you know what artificial hybridization is, right? Now you know what artificial hybridization is, and now you know how it is done. Right? No, you don't know how it is done yet, but you know why it is done. Right? You know why it is done. We are doing it to get favorable characters. We need favorable characters in the plant that we aspire it to be. Okay, we have a plant A which is ideal in every other means, but not in the means of disease resistance. So what will we do? We will cross A and B, and we'll produce a new hybrid C, and this C will have all the good qualities of A and one single good quality of B, and in that means the C will be a beautiful plant, an awesome mango tree with a lot of yield, with a lot of mangoes, with a lot of sweet mangoes, flowering every year and full of disease resistance. Right? It's the dream plant. Right? So this is the hybrid. Okay, the plant that we produce as a result of hybridization is known as the hybrid. Okay, so now you know what artificial hybridization is. So we do it for crop improvement. Of course, right, crop improvement, to improve yield of crops, of various crops, not just mango tree. Okay, so what is happening here? The pollen is crossed artificially, right? The pollen that we take is crossed artificially, and also the stigma is protected from contamination. What do you mean by that statement? Imagine, I've already told you that you have, okay, let me just erase that. Okay, you have two plants A and B, cool. You'll take the pollen of A, okay, we'll do it this way. It's not wrong if you do it the other way, but still, okay, this is the typical way, so we'll do it this way. You have a pollen of the plant A, and you have the stigma of the plant B. Cool. We'll take the pollen, the desired pollen, this is the pollen that we want, and we'll put it on the stigma of the plant B. You can do it either way, okay, it's totally cool if you do it anyway, but just listen to the process, that's what you need to get. Okay, so we'll take the desired pollen and we'll put it on the stigma of the other plant, and it will germinate over there, but in this time, you should not have any other pollens, right? Like imagine there's another plant called D. Okay, there's another plant called D, another mango tree, and this D is not a plant that we know. Okay, we don't know anything about D. It's a big question mark here. We don't know anything about D. So the pollen grain, if the pollen grain of D falls on the stigma of B, that would be a huge problem. Right, that would be a huge problem, because what if the pollen grain of D germinates? We want the pollen grain of A to germinate, but what if the pollen grain of D germinates? We never know which one is going to germinate. Right, so that's a huge problem. So in order to avoid that problem, the stigma should be completely prevented from any kind of contamination. Okay, there should not be any kind of contamination, or the pollen from outside, from an undesirable plant, should not be allowed to enter. Okay, so this is what you mean by the statement stigma should be protected from contamination. So now you know what artificial hybridization is. Now you know why it is done, but you don't know how it is done. Right, let's look at that as well. Let's look at the steps of artificial hybridization. So the first and foremost step is known as emasculation. What is it called? Just repeat the word with me. Emasculation. Okay, emasculation. So what is emasculation? Let me tell you. Imagine you have a bisexual flower. Okay, what am I saying? Repeat it with me. Imagine you have a bisexual flower. Okay, so emasculation is only done in a bisexual flower. You don't have to do it in a in a unisexual flower. It's only done in a bisexual flower. Okay, imagine you have a bisexual flower, and what is the problem of the bisexual flower? Self-pollination can take place, right? So if A was a bisexual, or A or B, whatever plant that we are choosing to pollinate, if it's bisexual in nature, so what will happen? We need the pollen grain of A to enter the stigma of B. Right, we need the pollen grain of A, just A. We don't even need the pollen grain of B. Okay, we only need the pollen grain of A to reach the stigma of B. But if B is a bisexual flower, then there are chances of the pollen grain of B getting onto its own stigma. So that should be prevented, right? So what will we do? We'll cut off the stamen of B. As simple as that. We'll cut off the stamen of B. So imagine you have imagine I'm drawing B, and you have the ovary, the stigma and the style and all that, and you have the stamens as well, right? You have the stamens as well. But if there are stamens and if there are anthers, what will happen? The pollen of the same plant of B itself will fall on the stigma, and that should not take place. In order for it not to take place, we'll cut the pollen of B. We'll cut the pollen of B. The stamen of B should be completely eliminated or cut. Right, this is what is known as emasculation. So it is only done in bisexual flowers. It's only done in bisexual flowers. Okay, it's only done in bisexual flowers. And what's the process? It's basically removal of anthers from flower bud. So this is usually done in a flower bud, because if the flower grows, there are chances of pollination to take place, self-pollination to take place. So we we take a bud, okay, we choose a bud, we don't pluck the bud, okay, we keep the bud in the plant itself. We just open it gently and we carefully remove the anther or the stamen with the help of forceps or something like that. We can even do it with hands if the flower is kind of big, but we'll usually do it with the help of forceps. Cool. So that's what is emasculation is. So this was the first step of our artificial hybridization. Now the second step is actually really easy. Okay, so now we have B, which is unisexual. We have turned into a unisexual plant by performing emasculation. Okay, so the B only has the female reproductive organ, right, the pistil. B only has the pistil. Now we cross it. Okay, we perform artificial hybridization. So basically the second step is artificial hybridization. Okay, the second step is the hybridization process itself, that is the transfer of the pollen grain to the stigma. Okay, that's the second process. The third step basically is the bagging. Okay, third step is bagging. So second step is hybridization, which we have already talked about, so I am not telling it in detail. It's hybridization. Okay, the second step is hybridization, and the third step over here is bagging. So as I already told you, the stigma should be prevented from contamination, right? So once the pollen grain from A has successfully reached the stigma of B, then it's necessary that we bag it. You know, that we cover it with some kind of plastic cover or butter paper or something like that and tie it. Okay, we need to cover it with a plastic bag or a butter paper or something like that, and we have to tie it so that another pollen grain, even if a pollen grain comes, it will be stuck outside. It will be stuck outside the bag. It won't be able to enter into the stigma. It won't be able to fall on the stigma, right? So this is the third step, or bagging. So this is done in emasculated flowers after hybridization. Okay, usually it can be of butter paper or plastic. Okay, I hope it's clear. I'll show you a picture. See, here you have a picture of a bud which is actually bagged, right? It's covered with a plastic bag or something, and it's tied, right? It's tightly tied. So even if any pollen grain comes, it will stay on the bag. It won't be able to enter the stigma. Okay, so this is all about artificial hybridization. Artificial hybridization ends here. Now what's our next topic? The topic that we've been waiting for, right? The fertilization. So pre-fertilization events are completely over, and now we are moving into fertilization. Let's look at what fertilization is. Double fertilization. That's your next section. Section 1.3 of your NCERT textbook, double fertilization. Why do you call it double fertilization? Let's see. Okay, so now what do we have in hand? I mean, where do we stand in reproduction right now? Okay, where do we stand? We should always recall where we stand. Okay, where did we reach? Okay, the male gametes reached the egg, near the egg, right? So in the ovary, so if this is the ovary, you had the ovule, right? You had the ovule, and the pollen tube have grown. And you have the egg. You have the synergids, right? You had the filiform apparatus, cool, and you also had the male gametes, right? You had two male gametes coming over from the pollen grain, right? You had two male gametes, the first male gamete and the second male gamete. You had two male gametes waiting to fertilize. Okay, so which of these two male gametes would fertilize? Okay, which one will be the lucky one to fuse with the egg, and what will the other one do? Only one fuses with the egg, right? Only one male gamete. So if you have a male gamete here, I hope you can't see what I'm writing. Okay, I'll redraw. I'll rewrite it. Okay, so if you have one male gamete, this one male gamete will obviously fuse with the egg. You only have one egg to fuse with, right? So you will fuse with one egg, and this will obviously form something. Okay, it will form something that we don't know yet. Okay, we'll study it. So you'll have one male gamete and you'll have one egg which will fuse together. But what will happen with the next male gamete? Where will it go? Is it so lonely that it's gonna die? No, it's not gonna die. It's gonna fuse with someone else. Who is there to fuse with? I mean, you've already studied the structure of female gametophyte, right? Who will the male gamete fuse with? The other male gamete, the second male gamete. Who will it fuse with? Let's look at the next slide. Okay, so here you have two headings, right? Syngamy and triple fusion. What are the two headings? The two headings are syngamy and triple fusion. Okay, in the case of syngamy, as I already told you, we have one male gamete fusing with the egg cell to form a zygote. So that's something that I told you before was a zygote. Okay, so the male gamete, which is obviously haploid, the egg, which you recall, is haploid again, right? Because it's all formed as a result of meiosis, right? So it's haploid. The male gamete is haploid and the egg cell is also haploid, and these haploid structures fuse together to form a zygote, which is diploid in nature. It's diploid in nature. Okay, I hope that's clear. And now you have triple fusion, the second heading, the triple fusion. And in this triple fusion, you can see that the second male gamete, which we thought was lonely, did fuse with someone. Who did it fuse with? It fused with the two polar nuclei. Yeah, you remember the polar nuclei, don't you? You had the polar nuclei at the center. You had two polar nuclei. They were besties. They were not ready to separate. you know, they were located in a central cell, right? You had the polar nuclei, right? So you have two polar nuclei, and both of them were haploid, right? So this male gamete, the second male gamete, goes and fuses with this two polar nuclei. Okay, so the male gamete is haploid, the two polar nuclei are haploid each, okay, and as a result you will get PEN, primary endosperm nucleus. Okay, what's the full form of PEN? It's primary endosperm nucleus. Okay, so as a result you will get the primary endosperm nucleus. I hope that's clear. And what will be the ploidy of primary endosperm nucleus? It's not written over there, I believe. Yeah, it's not written over there. You tell me, what will be the ploidy? Of course, if you look at syngamy, you have a haploid male gamete, you have a haploid egg cell. So M plus N, you had 2N, or a diploid zygote. Cool. Over here you have the second male gamete, which is also haploid, and you have two polar nuclei, which is haploid each. You have two of them, so it's basically N plus N plus N. Okay, what is it? It's N plus N plus N, which gives 3N. Right. So the PEN, or the primary endosperm nucleus, is triploid in nature. It's triploid, okay, because you have two polar nuclei which are haploid each, and you have a male gamete which is haploid, right? So these three fuse together to form the primary endosperm nucleus, and the primary endosperm nucleus will be triploid in nature. Okay, so the zygote is haploid, and the PEN, or the primary endosperm nucleus, is triploid in nature. Okay, keep that in mind, don't forget it. And now what will happen to the zygote and what will happen to the PEN? That's what our next topic is. So fertilization is over. Fertilization is basically a simple topic, and now you understood why it is called double fertilization, because there are two fusions happening, right? There is a syngamy and there is a triple fusion. It's called triple fusion because, again, three substances or three members are participating in triple fusion, right? So hence it's known as triple fusion. Clear? Okay, it's basically common sense. So you have syngamy and you have triple fusion, and as you have two fusions happening, you call it double fertilization. Okay, the zygote, which is formed as a result of syngamy, develops into the embryo, and the embryo develops into the new plant. So the zygote is the main character over here, right? Main character energy. So zygote is the main character over here, and zygote develops to an embryo. Okay, I hope that's clear. An embryo develops into plant later on. And you have the PEN, or the primary endosperm nucleus, right? So the primary endosperm nucleus will be lying in the central cell, right? So the central cell will now be called primary endosperm cell. It's nothing important, it's nothing confusing. Basically, you had two polar nuclei. They fused with the male gamete to form PEN, or primary endosperm nucleus. And the primary endosperm nucleus is now lying in a central cell, right? So now the central cell is special. So we'll call it with a special name, primary endosperm cell. I hope that's clear. Okay. And this primary endosperm cell, or the central cell containing the primary endosperm nucleus, develops into the endosperm. Okay, endosperm. Is that a new word for you? If it is, don't worry. We'll discuss what an endosperm is and what an embryo is in detail. Before getting into those details, I'll show you a figure. Okay, see, you have a figure here. What does this figure actually show? It actually shows the condition of a female gametophyte once fertilization has taken place. Okay, so the fertilization is over, and what's happening to the female gametophyte, or what's happening inside the ovule? Let's look. See, here, this is the chelazolant. Okay, it's basically inverted over here right now in this picture. So this is the chelazolant you have here, and this is obviously the micropylent. So in the chelazolant, you had three antipodal cells, right? So these antipodal cells degenerate. Okay, so these antipodal cells actually degenerate. It degenerates. Okay, clear? These antipodal cells degenerates, and you have the primary endosperm nucleus, right? The PEN, which is, as I already said, triploid in nature. And then the cell that the PEN lies in is known as the PEC, or the primary endosperm cell. Okay, which is also something that I've already told you. I've already explained. And then you have the zygote, right? Where will you be having the zygote? In the micropylent. Obviously, zygote will be produced at the micropylent because the egg was at the micropylent before, right? And the male gamete fused to the egg, right? So you will have the zygote at the micropylent, which will be diploid in nature. Okay, and those synergids, the two synergids that you had, which were the bodyguards of the egg, right, they'll degenerate because you don't have an egg anymore, you have the zygote and you don't need to protect anyone. So it's not actually in that way. Okay, but for your ease to remember, I'm telling it as, you know, a story, as a metaphor, just to remember. Okay, so you have the regenerating synergids because it's not needed anymore, right? The function of synergids was basically to protect the egg and to, you know, guide the male gamete towards the egg and all that with the filiform apparatus. Right now, synergids is also not needed after fertilization, so hence synergids are also degenerating. So this is how an endosperm, how an ovule or a female gametophyte would look after fertilization. Okay, is that clear? So fertilization is over. Now we'll be discussing what happens next, you know, what happens after fertilization. So you have post fertilization structures and events. We have already discussed about pre-fertilization structures, we have discussed about fertilization. Fertilization is also over, and now the last part of the chapter is the post fertilization structures and events, you know, things that are happening after fertilization. In that we'll talk about the embryo in detail, we'll talk about how it develops, the embryo develops, and we'll talk about endosperm and how it develops, and we'll talk about the seed and the fruit. Okay, we'll talk about all that in detail in the next video. Till then, work hard and stay safe. Bye bye.